Good morning. Well, it's morning when when I'm taping this, so good day. So today this is about chapter 3, and we're going to just be talking about 3.1, which is the methods of microbial investigation, 3.4, which is the additional features of the 6 I's, as well as 3.5, which is different types of media. So, in order to identify a microorganism, one goal of these procedures is to attach a name or identity to the microbe using the information that we gather from inspection and investigation. Then the identification is going to be accomplished through the use of various keys, charts, and computer programs that analyze the data to arrive at that final conclusion. So the first thing that we need to do is inoculate in a microorganism. And this is when the sample is placed into a container of media that will support the growth. And the media or the medium, same thing, right? And this could be a solid or a liquid. It could be held in tubes, plates, flakes, flasks, sorry, and sometimes even eggs. The delivery tool is usually a simple loop, a needle, a swab, or a syringe. Right now, when I'm recording this, this is in the middle of COVID, so we know a lot about swabs when you have to get COVID tested, right? But when we're going through the lab, et cetera, we're going to really talk about the different types of inoculation types or things that we can do. So then we gather information about these microorganisms. And so additional tests for microbial function and characteristics may also include inoculations onto specialized media that can determine biochemical traits, immunological testing, as well as genetic typing. And such tests will provide specific information that is unique to that specific certain microbe. So how do we collect specimens? Well, we collect them by trying to figure out what we're looking at, right? So if we're looking for something in a nose bacteria, well, obviously we're going to swab our nose. If we're looking for microbes that are in a water culture, we're going to get a culture from or a sample from water. So it could be nearly anything or a place on anything. So very common sources are bodily fluids, urine, blood, nasal swabs, etc. Foods. Unfortunately, there's a lot of lettuce and contaminations with E. coli, right? Water. My family has a place on a lake. We want to make sure that the microbial count isn't too high, right? Soil, plants, animals. But we can even look at things like icebergs, volcanoes, and rocks, and these can be sampled, soil samples, soil pores, right? So there's a lot of different things that we can sample in order to figure out what we want to find. Well, inoculated media are then going to be put into a controlled environment. And this is called an incubator. And this is going to promote growth. And during the hours or days of this growth process, the culture develops as the visible growth in the microbes in the container of the medium. And so when we put in a culture, an inoculated culture, it's probably going to be kind of clear, right? And then in a day or two, we're going to see that it's cloudy. Well, that cloudiness is going to be the microbes that have now grown in that specialized media. Well, then we have to look at them, right? And so this would be inspecting the microorganism. So cultures are absorbed for the macroscopic, remember macro meaning big, appearance of growth characteristics. Then the cultures are examined underneath the microscope for basic details such as the cell type and shape. And this can be enhanced through staining and through the use of special microscopes. Okay, so there's a lot of different microscopes, and your book goes into a lot of detail on those, and they're actually pretty cool. Okay. Um, Also, sometimes we can isolate the microorganisms. So we might do a streak plate and then say, okay, I want to look at just this one microorganism. And so then you can take and you can loop just that tiny little microorganism or that one colony, and it'll purify it a little bit more than just a generalized colony. So some inoculation techniques can separate microbes into isolated colonies that contain the single type of microbe. And this is very, very valuable for identifying the exact species of the microbe in the sample, and it also paves the way for making sure that we are working with a pure culture when we're looking at identifiable traits. So what are the 6 I's of culturing microbes? And they talk about these a lot, so this might be a great flashcard, right? I1 is inoculation, and this is the introduction of a sample into a container of media to produce a culture of observable growth. The second I is isolation, and this would be separating one species from another. The third is incubation, which is where you guys don't see that. Huh. All right, I just got an alert on my computer, sorry. So incubation is under the conditions that allow growth. Inspection looking at it, information gathering, or gathering our data, and then identifying, right? So those are your 6 I's. So make sure to flag those so that we can pay attention to those. So looking at each of them more specifically. Isolation. If an individual bacteria cell is separated from other cells and it has a space on a nutrient surface, it can grow into a mound of cells, or a colony. A colony consists of one species. So you can see that you can have the microscopic, macroscopic view, and then microscopic is down to the individual cell, right? You can plate out two different mixed culture, and then you can see that, you know, the yellow one's going to grow by itself and being isolated, and then the rods are going to be another, so that could be isolated and then recultured, so that we are looking at that one isolated species. So how do we do this? Well, this would be through streaking the plates. Okay, so this method works the best if your spreading tool, usually an inoculating loop, is re-sterilized after steps 1 of 3. And so basically what you're going to do is you're going to take your plate and you're going to sterilize your loop, and you're going to just look at that, you're going to pick up that one culture and you're going to streak it lightly across. Okay, great. Re-sterilize your loop, turn your plate just a tiny bit, and just overlap a little bit to streak it out a little bit more. So that's going to start thinning it out. Re-sterilize your loop, tilt another quarter turn, re-streak, sterilize, turn, re-streak. So you can see that this is a 4-step process, and you can see right here, your original inoculation is going to be very thick. But as we continue to spread it out and re-sterilize our loop in between each of the different streaks, you can see that this mess turns into isolated colonies as we streak it out. Okay, so this is a 4-part or a quadrant streak. Another technique is the pour plate technique, and this would be where you have your loop containing the sample. You're going to, you know, inoculate the media, and then inoculate, and then inoculate. So you just take one loop and re-sterilize, one loop, re-sterilize, right? So that you're, again, diluting it. Okay, so this is also called a pour plate or a serial dilution. So you're going to just, again, pour onto there, take one inoculate, and then inoculate and culture into the media, and then plate that and grow it. And then same thing, one loop, re-dilute, and then try to inoculate that. And so then you can see that over time, you're going to do a serial dilution. All right, another way is through the spread plate technique. And so this is where you would, you know, put your glob, like a puddle of your sample there. Then you're going to take your hockey stick and you just kind of spread it around, right? And so this would be just to take one small sample and spread it out, kind of like you would spread butter on a piece of toast, right? So this is going to be called the spread plate technique. All right, the next I is inspection. So if a single species is growing in the container, then you have a pure culture. But if there are multiple species, then you have a mixed culture. And we have to check for contaminants, which are unknown or unwanted microbes in the culture. Okay, so again, we're looking for all yellow guys here. If you see that there's some little white ones, then that's more than likely a contaminant. Okay. So again, different inspections. So you can see that you know, this looks like it's looking for the red culture, but then there's the white culture here. So that to me would be a contaminant. So you'd have to either re-isolate and re-streak, or ditch it, depending on what you're looking at. And then you can look when you're doing stabs. You know, if you have just a single color, fantastic. But if you are getting mixed cultures, then that would be not an isolated pure culture anymore. So there are a couple different ways to identify the microbe, and the cell colony morphology or staining characteristics are 2 of the biggest ones, right? So what does it look like? What are the colors, etc.? And then what types of stains does it accept? Also, we can do DNA sequencing to figure out what the genetic sequence is. There are also biochemical tests to determine an organism's chemical and metabolic characteristics, and immunological tests, so you can do serum tests, etc. Okay. There are different types of media, okay? So depending on what we're going to be looking for, we can use different types of media, right? So there's one that's kind of a really general purpose, but then there's other more like hemolytic proteins, right? You would want to culture them on a blood plate because hemolytic means lysing or lyticking. I made a verb, right? Blood, hemo would be the blood, right? So we need a blood agar plate to see if those microbes are going to be hemolytic. Okay, for an example, there's chocolate media, oh my God, there's a ton of different types. So media can be classified according to three properties. One is the physical state. Is it going to be liquid? Is it a semi-solid or is it a solid? The chemical composition, for instance, is it synthetic, which is chemically defined, or is it complex? And then the functional type, and this would be the general purpose. Is it enriched? Is it selective? Is it differential? Anaerobic, aerobic, transport, so you're going to look for microbes moving across it, right? An assay or an enumeration. So there's a lot of different types of media, okay? So that's awesome. The different physical states, well, this would be the physical states, right? So is it liquid, is it semi-solid, or is it solid? So a liquid is just the broth. It literally looks like chicken soup, chicken broth, right? No chunks in it, but it's just going to be a broth, and it turns from clear to cloudy when you have a microbial. Semi-solid, this is going to contain a solidifying agent. And then there's solid, which are those streak plates that we usually use. TSA plates are what I usually use the majority of the time, right? And so these are a firm surface for colony formation, and they contain a solidifying agent, usually agar. And this is liquefiable and non-liquefiable, depending on what type of microbe you're trying to grow. So again, you can see here are some liquids. This looks like it's a stab one. So it's, it was liquid and then it solidified, and then you stabbed your microbe into it so that you can just watch it grow in the colony, etc. So for agar, this is the most commonly used solidifying agent. It's solid at room temperature, but it does liquefy at boiling, and it does not re-solidify until it cools. These are actually kind of fun to pour. I know most of the time now we just buy them, but when I taught micro down in Kentucky, I got to pour them, and so I made it and poured it, and it was a ton of fun. So anyway, it provides framework to hold moisture and nutrients, and this is not digestible for most microbes, so it's just going to be like growing on top. So an example of the first one is that you would have a solid agar in a test tube, sterile and everything, and then you'd warm it up until it became liquid, and then you would pour it into your plate so that it can then solidify. Okay, but you have to make sure that top isn't off very long because you can invite contaminants, etc. So most common types of media are going to be your nutrient broth, and this would be the liquid medium that contains beef extract or a peptone, or the nutrient agar, which is the solid media that I talked about, which contains either beef extract, a peptone, and also an agar to solidify it. Okay. So the different chemical components of media are synthetic, which means that it contains pure organic and inorganic compounds in an exact chemical formula. Complex or non-synthetic, and this is where it contains at least one ingredient that's not chemically identifiable or definable. General purpose media, and this grows a broad range of microbes, usually non-synthetic, or enriched media, and this contains complex organic substances such as blood, serum, hemoglobin, or special growth factors that are required by fastidious microbes. So here is a fun little different ways of looking at media, right? So this is the growth of step pyrogenes that's going to show beta hemolysis. You can see that it's turning white around the microbe, and so that means that it's lysing or breaking apart that blood agar. This is a chocolate agar plate, kind of the same thing. So you have the dark media itself, and then when it's eating or digesting the nutrient media, then you're going to have the white or the clear areas around it. There are also selective and differential media, and this is where selective contains one or more agents that inhibit the growth of some microbes and encourage the growth of the desired microbe, or differential media, and this allows the growth of several types of microbes in displays visible differences among these microbes. And so, it's sort of like antibiotic resistance test, right? So you can inoculate your media with an antibiotic, and then put your microbe on top of it, and it will select against the ones that would be killed by the antibiotic. So the only ones that will grow would be the microbe that is resistant to the antibiotic that was impregnated in the media. And so that would be like a selective media, right? So looks like we are at the end of the chapter. So just a couple questions for you. So chrome agar contains several dyes that's used to diagnose your UTIs, and the patient sample is inoculated, and based on the color of the colonies, you can identify the pathogen. So chrome agar would be just what would it be defined as? Enriched, selective, differential, or complex. So think about it for a second. And it is differential, right? So because it differentiated between the different types of inoculates, right? Great. So some media can both be selective and differential, depending on what you're looking for. And again, the agar inoculation, you can see, the different colors that are growing. The color changes with pHs, changes, etc. A lot of different things. Okay, and then there's also miscellaneous media, and this would be, for instance, carbohydrate fermented media, and this is going to contain sugars that are fermented and converted into acids, and a pH indicator is used to show this reaction. So you can see that, that your control tube is like this. You can have a gas bubble that these are actually fun to work with. Gas bubble that forms if the media is being used, and also it changes colors. Okay, so there's a lot of really fun different medias that you can use, and we are going to play with a lot of them. All right, thanks, guys.